All right, class. One last thing I would like to add. So we did talk about spatial summation, where information is added up, like you guys shouting at me, Rupa, drink, or Rupa, don't drink. And then we have temporal summation, where it's only one of you talking to me and telling me to either depolarize or hyperpolarize. Okay, now one thing, and this is the last thing. When you have, or when we see positive charge entering and causing a graded potential, we call that epsp. So you have positive coming in, and this is causing local depolarization. This is epsp. And when, so again, this would be epsp, also called excitatory postsynaptic potential. Excitatory postsynaptic potential. EPSPs cause the soma to depolarize, and if it's depolarized enough, if it reaches threshold, an action potential will be generated at the trigger zone. IPSPs are caused by negative charges. So this one right here is an IPSP. IPSP is inhibitory postsynaptic potential, and IPSP moves the postsynaptic neuron further away from threshold, so it leads to hyperpolarization. Remember that in case of spatial summation, the postsynaptic neuron will add up all the EPSPs and IPSPs, and it'll decide and it'll see whether it can reach threshold or not. If it reaches threshold, you will have an action potential. No threshold, no action potential. So I compare EPSP to you shouting at me, drink. So drinking is an excitatory postsynaptic potential. I will drink, and my action of drinking can be compared to the actual action potential. If you tell me don't drink, that's an IPSP. I will keep the bottle down and the process will not continue. All right, so that was all my drawings, and I will do the PowerPoint super quick and end this chapter. Okay, so looking at the synapse, we do have neurotransmitters released. So let's look at the diagram. Let's look at it. So we have action potentials coming to the axon terminal. That opens the calcium channels. The voltage gated calcium channels open. Calcium enters the cell. Calcium causes the synaptic vesicles filled with neurotransmitter to exocytose the neurotransmitter in the synaptic cleft. Now there are terms like snare complex, docking, fusion. Don't worry about it. Just focus on exocytosis of neurotransmitter into the synaptic cleft. All right. So the neurotransmitter is a ligand, meaning it's a chemical that will bind to the to specific gates on the postsynaptic neuron, and that will open up those gates, and that will lead to entry of different ions. It can be an ion that carries positive charge or an ion that carries negative charge. So graded potentials. When a ligand-gated ion channel open, the membrane potential changes, right? There is local change in membrane potential depending on which ion is entering. Now read it carefully. When sodium and calcium channels open, that results in graded depolarization, and that is called EPSP, excitatory postsynaptic potential, because inside we are getting more positive charge. And in this case, we do see, let's look at the picture, we do see resting membrane potential, we also see threshold, right? And this blue thing is EPSP. It's a graded potential, excitatory. Now what happens if potassium channels open or chloride channels open? Well, when potassium channels open, we will lose positivity. And when chloride channels open, we will have chloride coming in. In either case, the membrane potential will become more negative and a state will be reached called hyperpolarized. And this type of graded potential is called inhibitory postsynaptic potential. Inhibitory postsynaptic potential leads the membrane potential to move further away from threshold. It moves the membrane potential to a more negative state, which is always further away from the threshold. All right, this is a summary slide. I'm not explaining this, pretty self-explanatory. You can figure this out, nothing special. Synaptic integration, we did talk about it. We talked about neuronal convergence, meaning when many upstream neurons are talking to one downstream neuron, or we have many presynaptic neurons and one postsynaptic neuron. That's convergence. And divergence is me talking to you guys. So one presynaptic neuron talking to many postsynaptic neuron. So neuronal convergence and divergence. Remember, when many presynaptic neurons talk to one postsynaptic neuron, the information is summed up, and that's called spatial summation. So right here. So it's talking about synaptic integration. We will talk about the different types of summation, spatial summation and temporal summation. In case of spatial summation, we have signals converging from multiple neurons at the postsynaptic neuron. So all the epsps and the ipsps are added together at the axon hillock, right? So in this case, we have two presynaptic neurons talking to one postsynaptic neurons. And at the axon hillock, all the information will be summed together, and if it reaches threshold, yes, there will be an action potential as shown by the red spike. That is spatial summation. Temporal summation is when there is only one neuron talking to the, or one presynaptic neuron talking to the postsynaptic neuron. So that's temporal summation. So in that case, only one of the upstream neurons will talk to the downstream neuron. And depending on how strong the stimuli are, it can either lead to threshold, and in that case, you will have an action potential or you might not have an action potential. That's the end of my lecture. Now, in a regular class, I extensively talk about the adrenergic receptors and cholinergic receptors. So I actually found two super good, like very good videos, which I'll post. And then on the study guide, if you go over the study guide for the nervous system, I will have questions for you. So you will watch those two videos and fill out the study guide. Those are short videos, I think 10 minutes each. So not too much, okay? And if you have any question, we will have a review session. I posted it Wednesday or Thursday of next week. So we can always, discuss if you have any questions on the nervous system then. All right, class. Thank you.